In this video, we will discuss about how this SOX2 helps in laryngeal cell survival. So before going into the topic, we need to understand why this SOX2 come into play. So SOX2 in the laryngeal cells is known to affect its migration and proliferation ability. Apart from that, in the previous video we have understood that in the soft matrices, the stem cells features goes up for the laryngeal cells, and the SOX2 has a role towards increasing the renewal ability of the stem cells. So from these two factors, they come to the point that SOX2 might play a role in laryngeal cell survival. So for that, we wanted to study how this SOX2, how the cells in the absence of SOX2 behave in the soft matrices. First, they prepared the SOX2 knockdown cells or SOX2 knockout cells using silencing RNAs against SOX2. And then they check the SOX2 mRNA expression levels using the qPCR and also using the Western blot. We can see that the SOX2 expression levels is very low in the mRNA using in the PCR results, qPCR results, and also the SOX2 expression levels were low in the Western blot, indicating that the SOX2 has been suppressed. SOX2 has been suppressed when they used silencing RNAs against SOX2. Thereafter, they wanted to do this apoptotic studies to check the SOX2 effect on the cell survival. So for that, they first cultured these laryngeal cells for 36 hours on a soft matrices. After that, they treat the cells with two different drugs separately that they did in the previous studies also, the cisplatin and 5-FU. And thereafter, they check the apoptosis using the flow cytometer and also using the CK assay in which they measure the absorbance. So when they check it using the flow cytometer, they are able to see that in the control there is not much difference they see between the SOX2 knockdown cells and SOX2 wild type cell. SOX2 is wild type cells here. Whereas for the cisplatin, we can see that in the case where SOX2 has been silenced using silencing RNAs, there is an increase in the apoptosis compared to the case where SOX2 is not been silenced for the cisplatin treatment. And similar results they have seen for 5-fluorouracil treatment also. Whereas in the case where SOX2 has been silenced, they are able to see increased apoptosis. And similar results they have seen when they check this cell viability using CGK assay, where the decrease in absorbance means decrease in viability. And we can see that for the cisplatin, when this SOX2 has been silenced, there is a decrease in the viability. And similarly for the 5-uracil treated cells, when the SOX2 has been silenced, there is a decrease in the viability. So these all results in together indicate that SOX2 might play a role toward this laryngeal cell survival. And from the evidence, it seems like in the soft matrices where it is known to increase in the soft matrices where laryngeal cells is chemo-resistance ability is been increased in the previous experiment, those abilities can chemo-resistance abilities can be reduced by targeting the SOX2. This is what they are able to come out from the results that are presented in this figure, and the role of this SOX2 towards not just laryngeal cell survival but its chemo-resistance ability. So this SOX2 is seems to play an important role, and from there and this is also in line with their hypothesis that they have tested, that where they are able to assume from the previous studies that SOX2 also helps in this cancer stem cell features in the soft matrices. And apart from that, this SOX2 about this laryngeal cells can directly affect its proliferation and migration abilities. So these two hypotheses seems to be true from the results that are presented in this figure about the apoptosis and of the apoptosis using two different drugs and their decreased chemo-resistance ability. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.